So first, what is DNA? DNA, or deoxyribonucleic acid, is the hereditary material in almost all living organisms. It's the instruction manual that tells cells how to build proteins which carry out nearly every function in the body. DNA is found in the nucleus of cells and also in mitochondria in small amounts. Now here is the structure of DNA. DNA is a double helix. Imagine a twisted ladder. The sides of the ladder are made of alternating sugar, deoxyribose, and phosphate groups, and the rungs are base pairs. There are 4 nitrogenous bases in DNA: adenine, A, thymine, T, cytosine, C, guanine, G. These bases pair specifically, a pairs with TC pairs with G. This pairing is held together by hydrogen bonds, which allow DNA to unzip when needed, such as during replication or transcription. Here is about DNA replication. Before a cell divides, it needs to copy its DNA. This process is called DNA replication. It ensures that each new cell receives an exact copy of the DNA. The steps include unwinding the double helix, unzipping the base pairs, using DNA polymerase, an enzyme that adds new complementary bases to each strand, resulting in 2 identical DNA molecules, each with 1 old and 1 new strand, semi-conservative replication. Now you need to know about genes and chromosomes. A gene is a segment of DNA that contains the instructions to make a specific protein. Humans have around 20,000 to 25,000 genes. These genes are organized into structures called chromosomes. Humans have 46 chromosomes, arranged in 23 pairs. 1 set comes from the mother and 1 from the father. Now, how proteins are made from DNA. The process of going from DNA to a protein involves 2 main steps. 1. Transcription. In the nucleus, the DNA sequence of a gene is copied into messenger RNA, mRNA. RNA differs from DNA. It has ribose sugar instead of deoxyribose. It uses uracil, U, instead of thymine. 2. Translation. The mRNA travels to a ribosome in the cytoplasm, where it's used as a template to build a protein. Each 3-base sequence, called a codon, corresponds to a specific amino acid. This process is fundamental to how DNA controls cellular function. Now you need to know about genetic variation and mutation. Every person has mostly the same DNA, but small differences, polymorphisms, make us unique. Sometimes changes in DNA called mutations can occur. These can have no effect, cause disease, for example, cystic fibrosis, sickle cell anemia. Contribute to evolution by introducing beneficial traits. Mutations can happen randomly or be caused by environmental factors like radiation or chemicals. DNA in forensics and medicine. Because everyone's DNA is unique, except identical twins, it's used in forensics to identify individuals, paternity tests, medical diagnosis of genetic disorders, personalized medicine, where treatment is tailored based on a person's genetic profile. Here is the DNA technologies. Several technologies have revolutionized biology and medicine. PCR, polymerase chain reaction, amplifies tiny DNA samples. CRISPR-Cas. 9. A gene editing tool that can precisely modify DNA. Gene sequencing determines the exact order of DNA bases. Gene therapy attempts to fix faulty genes to treat diseases. Now, what is epigenetics? Epigenetics studies how gene expression can be turned on or off without changing the DNA sequence. Factors like stress, diet, and environment can influence epigenetic markers. These changes can sometimes be inherited and have significant impacts on health and development. In conclusion, DNA is the blueprint of life. Its discovery and study have led to groundbreaking advances in biology, medicine, and technology. From understanding who we are to treating disease and engineering organisms, DNA is at the core of modern science. Thank you, and subscribe this channel for more biological topics. And do comment which topic you want to understand. I can make other videos for you with interesting animations.